So welcome everyone to the DMICE Research Conference, which is not at our usual time because, as often happens towards the end of the term, we have thesis and dissertation proposal defenses and actual defenses. And so today we have a dissertation proposal defense, which means that the candidate, Chris Klocke, will be telling us about what he's going to do for his dissertation, and his dissertation advisory committee is here. Quickly, by way of introduction, and most people know Chris, but maybe if you don't, he's a PhD candidate and an NLM trainee in the Bioinformatics and Computational Biomedicine program. He has a Bachelor of Science in Biology from American University and worked in biotechnology development before starting our program, and currently does research in the genetics of cancer and mechanisms of immunotherapy resistance. I'd also like to call out Chris for his service to the program in serving on the curriculum committee and also representing OHSU on the program committee of the NLM trainees meeting, which many of us will be attending next month. So the usual procedure here is that he gives a public presentation, and then we open questions to the public, to everyone who's attending, and then the public is dismissed, and the Dissertation Advisory Committee may have some additional comments and questions for him after we all leave. So I will turn it over to Chris.
Thanks for that introduction, Bill. As you mentioned, I'm going to be talking about my approach for identifying gene regulatory network patterns affecting treatment outcome for a particular class of cancer immunotherapies. At a broad level, the clinical need driving this sort of research stems from the massive morbidity and mortality inflicted by cancer. This category of diseases is a leading cause of death globally and kills 600,000 people per year in the United States alone. Current treatments are inadequate to address this clinical challenge, and further research is needed to resolve this gap. Tumors are made up of a heterogeneous population of cells, which poses a key hurdle to achieving durable treatment response. A majority of malignant cells within a tumor may be sensitive to a chosen therapy, but the treatment will act as a Darwinian selective pressure, selecting for a small resistant subpopulation of cells that expand and drive disease recurrence. In the past decade, we have witnessed the development of a range of technologies that can profile various molecular processes in a tissue sample at the single cell level of resolution. This is particularly useful when we're trying to understand and study the tumor heterogeneity that drives treatment resistance. Single-cell RNA sequencing is a technology of particular interest because it allows researchers to obtain a gene expression profile for each of thousands of cells within a sample. We can then study numerous cell types and cell states that may be present. One limitation to consider when using these single-cell assays is sparsity. While we can get a single-cell level of resolution, some signal will be lost due to biological and technical sources of dropout, and we will not get an accurate quantification of every gene in every cell. When studying biological systems, the classical approach is to consider one gene at a time and to determine its function or disease relevance. However, it has become clear that in order to reach a deeper level of understanding, we need to take a more holistic system-level view. We can consider something like signal transduction pathways consisting of various protein-protein interactions as being part of this system-level view, but my research really focuses on the gene regulatory networks that control the expression of genes within a cell. If we think of a gene which may code for a transcription factor, that transcription factor may then interact with different regions in the genome in a way that activates or represses the expression of other genes. Some of these other genes may be transcription factors and may code for transcription factors themselves, which then have other downstream targets. And in this way, we have these interlinked systems of regulation of gene expression. Now, these networks are typically very carefully controlled, but they can become dysregulated in cancer. And it's important to understand how these GRNs become dysregulated to fully understand mechanisms of treatment resistance. One convenient side effect of taking a gene regulatory network level approach is that it helps to address this sparsity limitation of single cell data that I mentioned, since we are focused on sets of genes that are co-regulated rather than individual genes. We can think of a so-called regulon, which consists of a transcription factor and its direct downstream targets, as sort of the basic functional unit of a gene regulatory network. These so-called regulons can be inferred with various computational approaches that I will discuss in more detail. The treatments that I'm interested in fall into a category known as cancer immunotherapies. Now, the question that really drives the development of this class of therapies is, can we somehow harness or modulate the patient's own immune system in order to treat cancer? Part of the appeal of this is specificity, since a lot of existing approaches to cancer, are damaging to healthy tissue, which leads to side effects and are very hard on the patient. So the ability to target malignant cells while leaving healthy cells intact is very appealing. And as you can see in this image on the left here, we have in green, we have a cytotoxic T cell that is in the process of destroying a malignant cell. One promising class of cancer immunotherapies that I'm going to focus in on a little bit here is known as immune checkpoint inhibition. In healthy patients, the immune system can often recognize and destroy cells that are dysregulated and may develop into a tumor. However, certain inhibitory signaling pathways known as immune checkpoints can downregulate or deactivate this cytotoxic immune activity. One particular immune checkpoint consists of the proteins PD-1 and PD-L1, which we can see in the figure. Tumors can take advantage of this type of inhibitory signaling in order to evade destruction by the immune system. In the past decade or so, researchers have developed drugs to interfere with the signaling of these inhibitory pathways. They've developed monoclonal antibodies known as immune checkpoint inhibitors that bind to these immune checkpoint proteins and interfere with their inhibitory signaling. And in this way, they can reinitiate the immune response against the tumor and cause it to recognize these malignant cells once again. Inhibitors of PD-1, PD-L1, and CTLA-4 have become FDA approved for several different cancer types and have generated particular interest in recent years. Now, these drugs are highly successful in a subset of patients, which is part of what has generated this interest. They can lead to years of disease-free survival in patients with otherwise untreatable, late-stage metastatic disease who have no other viable options among existing therapies. However, that being said, most patients do not respond. And what's more, the mechanisms by which some patients respond and others don't is not fully understood, and we can't predict with high accuracy ahead of time who's going to respond and who will not. Since the response to these therapies is based on an interaction between multiple cell types, primarily the malignant cells and the T cells, we need to understand both tumor intrinsic and tumor extrinsic factors that may be involved in treatment response and resistance. So we need to understand the malignant cell populations that may be capable of driving resistance to these treatments. We need to understand T cell states that affect treatment outcome. And importantly, particularly for my first aim, we need to understand the influence of other immune cell types in the tumor microenvironment. I now want to take a minute to mention a cellular process that has caught the attention of researchers in recent years, known as T cell exhaustion. If you look at the top part of this diagram, kind of starting over on the left here, we can see the progression of T cell states in the case of a normal infection. So when a naive T cell encounters its specific target, it becomes activated into an effector T cell, which can then work to clear the infection. A subset of these activated T cells will be retained as memory T cells, ready to be quickly reactivated upon a second exposure to their target. Now, if we consider the bottom part of the diagram, we can observe a different process that occurs when T cells are consistently exposed to their target over a long period of time without eliminating it. This could be due to chronic infection, such as hepatitis C, or a tumor that can't be fully eliminated by the immune system. This chronic stimulation can drive T cells through a series of cell states known as exhausted. And in these exhausted T cells, they have, as you can see on the top right, they have reduced effector function. It's not absent, but it's much less. They express a variety of these immune checkpoint proteins that I described previously, and they have kind of distinct altered gene expression and chromatin accessibility profiles. While this research is still at an early stage, and this is just a model of our current understanding, it appears that there are a series of cell states that exist along a continuum of T cell exhaustion. These range from progenitor exhausted T cells that have stem cell-like properties to more terminally exhausted T cells. Whether immune checkpoint blockade therapy can reverse exhaustion in some cells, or if it causes progenitor cells to differentiate and kind of temporarily have more effector function, is not really clear. However, there is some evidence to support that patients that have a greater proportion of progenitor exhausted T cells to terminally exhausted do show better response. But as I mentioned, this is all just kind of a model of a recent understanding that's come in the past couple of years, and more work is needed to really sharpen this understanding in the context of human tumors. There has been some work that's been done using single-cell RNA sequencing and other methods in order to characterize both the tumor extrinsic and tumor intrinsic factors that drive treatment resistance. The tumor extrinsic factors, such as T cell exhaustion and the tumor immune microenvironment, there has been some work to characterize T cell exhaustion, but there are a few limitations to the work that's been done. To start, most of the work that's been done to characterize this has been done on a particular viral infection known as LCMV, so mice that are chronically infected. And many cancer immunologists have stated that these findings may or may not generalize to the context of human tumors, and it's important at this point to kind of move beyond that model in order to determine what's actually going on in the context of human tumors. The existing approaches have been, for the most part, overly gene-centric rather than kind of taking a holistic system-level view. They've been mostly focused on the T cells themselves rather than malignant cell populations and other immune cell types. So in order to address these limitations, these shortcomings, we need a better characterization of T cell exhaustion, specifically in the context of human tumors. We need a system-level understanding of the gene regulation patterns that are involved. We need a comparison to be able to compare and contrast between what's going on in the case of human tumors and human chronic infection, so we can determine what results generalize from the case of human, from the viral case to the tumor case, and which don't. And then, importantly, particularly for my first specific aim, we need a better understanding of what contributors in the tumor immune microenvironment could potentially be driving this T cell exhaustion process. I will address each of these with my approach. As for the tumor intrinsic properties of the malignant cells themselves that are associated with driving response or resistance to these drugs, we can think about this first in kind of the simplified case analogous to a targeted therapy, where we have many cells that are sensitive and a subpopulation that are resistant. And when we apply the drug, the resistant population is selected for and then expands. However, in the case of immunotherapies, it's potentially a little bit different than this because, as I mentioned before, the mechanism of response is based on an interaction between multiple cell types, not just whether or not the cancer cells themselves are responsive. So we could actually have cases where there's activity in a subpopulation of malignant cells that actually prevents the destruction of or reduces the destruction of any of the malignant cells within a tumor. And there's been some work using single-cell RNA sequencing that's shown some expression activity that causes a reduction of infiltration of T cells into the tumor, based on this tumor intrinsic signaling. However, this is really just kind of preliminary work and a much greater understanding of what tumor intrinsic activity can actually contribute to immune checkpoint blockade resistance is needed at this point. So in order to address kind of these gaps in our current understanding that I've described up to this point, it kind of brings us to two questions. So the first question is, what activity in particular immune cell types in the tumor microenvironment could be helping to drive this T cell exhaustion process? So our goal is to identify these contributors. The hypothesis here is that I can use sample level ordering based on the trajectory of CD8 T cells in order to identify these microenvironment drivers of exhaustion. And I'm going to talk about what I mean by that in much greater detail. The second aim I propose, well, the first aim is more focused on immune cell types. The second aim is focused on the tumor intrinsic factors, so the activity in the malignant cells. The question here is, can we identify what activity is present prior to therapy that contributes to resistance, but then also how this expression shifts and potentially changes over the course of treatment, particularly in patients who are resistant. So we want to identify clonal evolution and gene regulatory network changes particularly in resistant patients. And the hypothesis here is that I can use inferred copy number variation to track subclonal populations of cancer cells longitudinally across ICI treatment. And again, I'm going to explain exactly what I mean by that in greater detail. The first aim, this is just an overview. I'm going to walk through each of these steps. In the first aim, we're going to be identifying tumor immune microenvironment drivers of T cell exhaustion by walking through a series of steps. To start, I'm going to take single-cell RNA sequencing data from samples from patients with various skin cancer tumors that have not yet been treated with immune checkpoint inhibitors. I'm also going to, as an addition to this, I'm going to be taking immune cells from some patients who have a chronic viral infection with HIV. The main part of the analysis focuses on this tumor sample data. First, I'm going to use a cell cluster annotation process in order to identify various cell types and cell states. So as we can see on the right, the malignant cells are going to tend to group together. If we were to break down these clusters of malignant cells, the cells from each sample, they would kind of cluster out by tumor of origin based on their intertumor heterogeneity and their specific mutation profiles. However, when we look at the immune cell types, they tend to group together based on their expression profiles into their specific cell types and even states within cell types, with each cell type consisting of cells from a variety of samples. So so in this way, we're kind of able to analyze them all together, which is nice. So based on using known gene expression markers that identify these cell types, I'm going to select out the CD8 T cells from each sample. I will then take these CD8 T cells and perform a trajectory inference process. using one of several established algorithms, this T-cell, this trajectory inference process uses the gene expression profiles of cells to kind of put them into this pseudotemporal ordering of, a continuum of shifting cell states here. So one trajectory inference algorithm that has shown some success is known as PAGA. So this is kind of a figure, but there are other, like Monocle 3, there are other leading methods that are based on kind of this similar idea, where we have a k-nearest neighbors graph here, where cells that have broadly similar expression profiles will be linked as close neighbors here. And then we have these different clusters of cell states shifting from one to the next along this continuum that gives us this this pseudotemporal ordering, of this trajectory of cells. So in this way, I'm going to be using this algorithm in order to put these T cells in order. We might expect to get some naive T cells, effector T cells, and then ideally, I'm hoping to see, a series of progressively more exhausted T cell states. And I won't know exactly what sort of lineage topology to expect until I actually run the algorithm, whether it's going to be, an actual continuum with the intermediate states sampled, or if I have, more discrete cell states. But really the point of this here, if I was just interested in the expression of the CD8 T cells themselves along this trajectory, I could kind of just stop here. But since I'm interested in identifying other activity in other immune cell types, I kind of have to take it up to the sample level. So at this point, I'm going to use that cellular level ordering of the CD8 T cells to perform an ordering at the sample level. So just to kind of take a simple case here, if we imagine the exhausted T cells from a particular sample will be distributed somewhere along this this exhaustion trajectory, and they're not going to be distributed in exactly the same place for each sample. So in this way, we can place the cells in order, using various methods that I can discuss if we have time, to place these samples in order based on the overall degree of T cell exhaustion in that sample. Now, once we have this information, just a step away from specifically the CD8 T cells for a minute, I'm going to be performing gene regulatory network inference on a variety of immune cell types. In order to do this, I'm going to be using, there's several algorithms. One such algorithm known as scenic, the way it works is it looks at transcription factors, and then it looks at other genes that are highly co-expressed with that transcription factor. And that sort of gives you a first pass of what might be regulated by that transcription factor. That is then further enriched based on the presence of transcription factor binding motifs in these potential directly regulated targets. Then at that point, we can get these regulons I discussed with the transcription factor and its downstream targets, and we can quantify the degree of activity of that sort of gene regulatory network subnetwork at a higher level. So at this point, I'm going to perform gene regulatory network inference to identify these immune cell type specific regulons. And then once I have this information paired with this sample level exhaustion ordering that I discussed in the last step, we're going to put the two together. So just to take the simple case, I'm going to be looking for an association between immune cell type specific regulons and this sample level exhaustion ordering. To take the simple case of a monotonic relationship, we can just use kind of a rank-based non-parametric correlation to look for regulon activity that kind of tracks with this exhaustion. In the case of non-monotonic relationships, there are other statistical frameworks that I can use to identify groups of regulons that are active at particular points in this T cell exhaustion trajectory. At this point, once I've identified gene regulatory subunits, these regulons that are associated with T cell exhaustion in the tumor immune microenvironment, I can then start to put the pieces together and analyze this from a more system level. So at this point, if I identify a transcription factor that has a set of downstream targets, one of those downstream targets may code for, or may be a transcription factor that has its own set of downstream targets. And in this way, we can start to establish this, this hierarchy of these regulatory relationships. And then once this has been done, I can perform a gene regulatory network analysis to look for things like the degree of flow through particular nodes in the network, and if we have an activity change in a particular transcription factor, what percent of the variance of that is being explained by the activity of some upstream regulator, and we can start to get this more holistic picture of actually what's going on in this regulated system. Now, I mentioned briefly that I'm going to be including some data from chronically infected patients along with this in the trajectory. And this is just as an addition to this aim. This is going to allow me to compare and see what T cell exhaustion expression activity in these CD8 T cells is shared between the chronic infection case and the tumor case, and what is tumor specific. And the important thing is that this will help us start to be able to figure out what results from the chronic infection case actually generalize to the human tumor case and which do not. So just to recap here, I'm going to start with this single cell data. I'm going to use cell cluster annotation to select that the CD8 T cells. I'm going to perform trajectory inference to get a pseudotemporal ordering of these CD8 T cells that are progressively more exhausted. I'm going to use that cellular level ordering to get a sample level ordering in order to rank the samples based on their overall degree of T cell exhaustion, and then look for these gene regulatory subnetwork modules that are associated with this exhaustion ordering. And then I can perform the system level gene regulatory network analysis. Upon completion of this aim, I expect to have found several things. I expect to have identified some of these immune cell type specific regulants that are associated with T cell exhaustion. I expect to potentially have identified some of these gene regulatory network relationships, and to have a greater understanding of this comparison of exhaustion between tumors and the case of chronic infection. Some limitations to this approach are the uncertainty inherent to the computational inference of these gene regulatory networks. The fact that this exhaustion process still is not fully understood, so there's no underlying ground truth here, although we have some understanding from the literature to guide us here. And as with any correlation-based, computational analysis, there's the potential for confounding factors here, although I will be taking some steps to help mitigate that. And with that, I want to kind of shift gears to my second specific aim here, which focuses on tumor intrinsic factors, so expression activity in the malignant cells themselves that is associated with resistance to immune checkpoint inhibitor therapy. This is sort of an overview here, so I'm going to be walking through a series of steps, and then again, I'll recap at the end. To start, I'm going to be taking single-cell RNA sequencing data from some skin cancer tumors. And here we have data from responders and non-responders to therapy, and also paired data from paired samples pre and post-treatment. So this allows for a longitudinal analysis where we can see change over time. The central part of this aim is really based on the computational inference of copy number variation, which allows us to identify genetically distinct subclones that can then be tracked longitudinally throughout treatment. So just for a minute here, if we think about a subpopulation of cells, based on their expression before treatment, how do we know which cells post-treatment, these cells correspond to? How do we kind of do this one-to-one mapping to actually be able to track cells longitudinally across therapy? And one way to do this is to actually explicitly have genotype information. But since we're working with just single cell expression profiling here, this is why I'm adding in this, this use of computational inference of copy number variation. So there's an algorithm known as Honey Badger, which basically the way this works is it takes two pieces of information as input in order to predict if large stretches of chromosomes are amplified, deleted, or if they are kind of just left intact or copy number neutral. And the way it does this is it uses, so two pieces of information. The first information is gene expression. So is the abundance of expression increased along this section of the genome, or is it reduced or absent, which can indicate deletion? And then in addition to that, we also use this allele balance information from transcribed SNPs. So if we think about just heterozygous sections of the genome where we have different alleles and different SNPs present, then when genes are transcribed, these differences show up in the mRNA transcripts and then in our sequencing reads. So in this way, we would expect to see something around 50-50 allele balance in normal heterozygous regions. But if we see something more like 80% one allele and then 20%, that could indicate that one is amplified. Or if we have a large region where it's just not heterozygous and there's one missing, this could indicate some sort of deletion or loss of heterozygosity. So in this way, we can use this computational method to infer copy number variation that then allows us to define these genomically distinct subpopulations of cells within a tumor, which then allows us to track longitudinally. So this subpopulation prior to treatment corresponds to another subpopulation following treatment. I first just want to direct your attention to this figure on the right here. So this comes from a paper a couple years ago that looked at clonal evolution and transcriptional reprogramming in the case of chemotherapy. And the nice thing here is that they had single cell expression and single cell genotype information. And they were able to identify that there were these multiple genetically distinct subpopulations of cells prior to treatment. Most were resistant and were just eliminated by the therapy, but there was this subpopulation that was resistant and was selected for and expanded following treatment. However, the important thing is here, in addition to just discovering this clonal evolution, they also saw transcriptional reprogramming. So the expression profiles of these cells post-treatment didn't resemble the expression of any of the cells pre-treatment, showing that there was this genetically distinct subpopulation that was capable of resistance, but then its expression profile shifted in response to therapy. So kind of taking a conceptually similar approach to the case of immunotherapies, we're going to be looking for, first in the case of patients who respond, we might expect to see clonal extinction. So subpopulations of malignant cells that are reduced in frequency or eliminated over the course of treatment. However, in patients who are resistant, we would expect to see either the kind of clonal persistence or clonal expansion following treatment, which we can identify using this inferred copy number as a link between the two. Once that's been performed, focusing primarily on the patients who do not respond to therapy, I can perform this gene regulatory network inference that I mentioned in the first step to look for these regulons in pre and post-treatment malignant cells and start to quantify their change in activity. So, for example, do we see a subpopulation of malignant cells expressing a regulon that is very much upregulated or downregulated longitudinally across therapy during resistance? In addition to this, we can look at regulatory reprogramming. So while a particular regulon might be defined by a transcription factor and have a certain set of targets prior to therapy, the same transcription factor may have a mostly overlapping but slightly different set of direct targets after therapy due to things like different levels of chromatin accessibility and other things. So we can see this actual rewiring of the regulatory relationships of the gene regulatory network here. After completing this step, similarly to AIM-1, I can reconstruct the gene regulatory network hierarchy here to look at some of these regulatory relationships at a whole system level. And I can analyze these expression contexts, or these expression changes in the context of the gene regulatory network. So again, we can picture a particular transcription factor, whose activity changes longitudinally across treatment during resistance, but then potentially that's being influenced by one or more upstream regulators of that, and we can start to see that process unfold. And this has been shown to be, this type of analysis has been shown to be useful, where we might have multiple targets of immune checkpoint inhibition that are actually both controlled by the same upstream regulator. So this allows us to identify that sort of thing. Just to recap here, I'm going to start with single-cell RNA sequencing data from responders and non-responders with this paired pre and post. I'm going to use inferred copy number to identify these subclonal populations and then track them longitudinally across treatment. I will then perform a clonal evolution analysis and look for transcriptional and regulatory reprogramming that occurs particularly in the case of treatment resistance. And then I will perform a gene regulatory network analysis and be able to analyze this system as a whole. Upon the completion of this aim, I expect to have identified several things. I expect to have identified these patterns of clonal evolution, various patterns of transcriptional and regulatory reprogramming associated with treatment resistance, and some of the gene regulatory network relationships between these transcription factors. Some limitations of this approach include, some uncertainty inherent to the computational inference steps, as well as a limited sample size. On completion of these two specific aims, I expect to have contributed to the field and helped fill some of these gaps in several ways. I will, with my first aim, have identified immune cell type specific regulons that are associated with this T cell exhaustion process. And then with my second aim, identified changes in malignant cell gene regulatory network activity across this ICI resistance process. I will have created two computational pipelines that can be applied to novel biological questions. And then using these pipelines, I will be able to enable further analysis. So these can be applied. The pipeline from the first aim can be used for the discovery of cell extrinsic contributors to cell state change. And the pipeline from the second aim can then be used for longitudinal tracking of cell subpopulations using expression data alone, where genotype information is not available. And then to just take a more kind of long-term translational view and think about some of the more clinically relevant work that this research could support and enable. We can think about the future potential for targeting the tumor immune microenvironment in some way to improve the response rates to immune checkpoint inhibition therapy. And additionally, kind of in the case of the tumor intrinsic factors, we can think about the potential for targeting tumor intrinsic programs driving resistance. And in this way, by modulating the activity of either the malignant cells or the tumor immune microenvironment, potentially we can take this class of therapies that is highly successful just in a small subset of patients, and to really expand the number of patients that have the potential to benefit from these types of therapies. In order to complete this work, I plan to get started this summer on data processing and various normalization steps working with the data. I will then move into the early part of the analysis, performing some inference steps and start building these computational pipelines. We'll then get to a point where I'll start to get some results and I can analyze them and write them up into some manuscripts, which will lead me to ultimately the point where I am ready to defend my dissertation. At this point, I would like to thank my advisor, Guanming Wu, who has worked with me to help develop this research proposal, as well as the rest of my committee, Shannon McQueenie, Amy Moran, and Andrew Adey, who have worked with me to help put together this proposal. I'd like to thank the DMICE, all the students and fellows, who have really given me a lot of useful feedback and support throughout this process. As always, the DMICE faculty and staff who are really helpful, as well as a variety of other people who have, given me useful feedback as I've developed this research approach. And I'd like to just acknowledge my funding from the National Library of Medicine grant. And with that, I would like to open it up for some questions.
Great, thanks for your presentation. People can either unmute and ask questions or type into the chat. Any questions for Chris?
Hey Chris, great talk. I had a question and I probably just missed it. Can you explain what a pseudo-temporal?
Yeah, pseudo-temporal ordering, sure.
Yeah.
Yeah, I was kind of using two different terms there, which might have made it unnecessarily complicated. But basically, the idea of these trajectory inference algorithms is that, if we have a series of cell states that may be similar, but they're kind of transitioning from one to the next, the idea is that cells that have, very similar expression profiles are kind of maybe in the same or a similar cell state. And then we can kind of use, this transition from one cell state to the next. We can infer this computationally kind of based on this k-nearest neighbors graph type approach. And in this way, we can see, maybe we have naive T cells that then transition to effector T cells that then transition to some other cell type. So we can start to see, we can perform computational inference using expression data as input to start to kind of see how these cell states progress from one to the next. So that's kind of broadly speaking what these trajectory inference algorithms do. And then pseudo-temporal ordering is kind of just a jargon term of art that describes kind of this sort of path or trajectory that is inferred by these algorithms.
Okay, cool. Thank you.
Hey Chris, it's Jennifer. Great presentation. Here, I can turn my video on. So you said that you are, for your second aim, you're inferring the copy number based on gene expression and allele expression. But you also said that like after treatment, the gene expression of a particular population can change, right?
Right.
So sorry if I missed it, but did you talk about how you're doing that mapping of, if you're basing the copy number on gene expression and then gene expression changes potentially, how are you kind of reconciling that?
Yeah, that's a great question. Let me just go to the slide here so I can. Right, so we want to be able to track kind of from one population to the other. So, right, so the approach is based on this computational inference of copy number variation, like you said, and the idea is that the expression profiles of these subclones will show these genomic alterations in ways that can actually be detected to identify these subpopulations of cells, but that we might see small changes in the activity of these gene regulatory networks that can then be identified. So the thing here is these profiles kind of the inferred copy number, they don't have to be exactly identical for this to work. If we think about this kind of like in a like a phylogenetic tree where we have these multiple subclonal populations, even if two subpopulations are a little bit different, or like you said, the expression changes that we're trying to detect with the analysis, I guess really the heart of your question is, couldn't that cause you to infer that maybe they're not actually in the same subclonal population, which could cause a problem. But as long as they are similar enough, we can still kind of classify them as being in the same subclonal population. And just kind of in addition to that, some treatments, like something like a chemotherapy, you would expect it to induce additional mutations, which could cause differences before and after. But that's not really a concern here for two reasons. One of which is because there's evidence to suggest that immunotherapies actually don't really induce many or any genetic alterations through the course of treatment. But then kind of just to tie that back to your original point, even if there is some difference in either the actual copy number variation or our inference of the copy number variation based on the expression, as long as the overall inferred copy number profile is broadly similar enough to allow us to kind of identify these subpopulations of cells as more similar to each other than than other malignant cell subpopulations, then this should still work. So it's a really great question. Thanks.
Yeah, that makes sense. Nice job, Chris. This is great.
Thanks.
Chris, I'm going to follow up on that question just to ask you, can you think of gene regulatory networks that you may propose to regulate some of the subclone outgrowth if it's not through robust copy number changes due to a carcinogenic or some sort of environmental stress, like chemo? Can you think of what sort of gene regulatory networks you may expect to see under immunological pressure?
Yeah. I just want to make sure I'm understanding your question. So are you really getting at the link between the copy number changes themselves and the activity of the gene regulatory network, or are you asking about like specific, like biologically, what specific types of processes might be involved in this?
Biologically, because I don't think you're
Okay.
going to see, the biological processes and gene regulatory networks that you think would be changing.
Right. I mean, at this point, it would be kind of a guess. I could base that on what I've seen in past research. Like for example, there's some data in single cell data of analysis of melanoma that shows these kind of T cell exclusion programs where there's expression in malignant cell subpopulations that leads to less infiltration of T cells and other immune cells into the tumor microenvironment, which could make the tumor as a whole less responsive to immunotherapy. If I were to kind of think about what other types of things we could discover here, I mean, this could be anything that affects the presence or absence of immune cells in the tumor immune microenvironment, the degree of inflammation, inhibitory signaling, the things that allow the tumor proliferation. It could really be a variety of different things. And so it's at a certain point I'm going to have to see what I find and then see if that makes sense in the context of of what is known. But yeah, it'll be interesting to see what I find there.
Any other questions for Chris?
Hey,
Okay. Oh, go ahead,
thank you.
Ben. Yeah.
Chris, I really appreciate your explanatory skills. It really, I get a lot out of your presentation. Um, cancer immunotherapy has some pretty big problems with also sort of collateral damage, even if it's not as all comers as chemotherapy, right? So the endocrine system is really famous for getting hit accidentally or a collateral for immunotherapy. So to tie that concern into this, is there some way that you might be considering either now or in the future the adaptive functions of T cell exhaustion in a way that, you know, it might be positive, or there might be problems if you reverse those exhaustion processes? And so I just want to ask that broad question. Does that factor into how you're looking at this?
Yeah, absolutely. That's such a great point, and that's something that I've thought about a lot. And I, just it was a little bit of out of scope to go into it in the main talk, but I'd be happy to talk about it. I'm trying to figure out which slide. Yeah, actually, I'm going to go back to, so we have these immune checkpoints, and if we think about this from an evolutionary perspective, these aren't just there for no reason. It's there's this balance between if we have an immune system that a patient with an immune system that's not working enough, then potentially that person is going to be at greater risk for disease from different pathogens, and also potentially cancer from less immunosurveillance. But then if we kind of just, it's not, the immune system isn't a pure positive. If we just endlessly ramp it up, we get autoimmunity, which can be just as devastating. And so it's proposed, pretty plausibly, I think, that these immune checkpoints evolved as a kind of a prevention of excess immune activity and excess cellular damage. And so this T cell exhaustion process, many people have proposed that if these T cells are just being consistently stimulated and they're they have this consistent effector activity, then you mentioned collateral damage. It's just it's too much cytotoxicity to the surrounding healthy tissue, and so that this might be an evolved way to kind of strike a balance between the two. And then to just pull it back to kind of the clinical point where you really started, yes, there is cytokine release syndrome, and many of these patients who get immunotherapy, I think often in patients who do show a response too, you get these cytokine release events, and you get this autoimmunity, which is that in and of itself can be a huge problem. So kind of trying to figure out why some patients experience certain potentially very harmful side effects from these therapies, and kind of how to either predict those or mitigate those is really a whole area of research unto itself that would be really interesting to dive into. But that's I always appreciate when you bring in the clinical perspective, and it's important to think about not just these aren't a silver bullet, not just the potential benefits of an approach, but the way that they could actually harm the patient. So it's a really great point. Thanks.
Thanks, Chris. Great work.
Chris, I don't know if I should save all my questions and comments until we meet, but I just want to highlight Ben's point here, because that might be a really interesting data set for you to look at if it's available down the road. I know we're tracking these immune-related adverse events in these patients. As immunotherapists, we expect them, and so we're recording them and we're studying them. But it might be very interesting to look in the data sets in which there is, rashes, patients developing all these rashes, and are they sequencing and doing single cell sequencing on the T cells in the non-tumor tissue that would provide insight in the mechanisms of reactivation, right? Because those were cells that were not causing pathology until checkpoint blockade was introduced, and now they're pathogenic, and so their function has been released. And so overlaying those data sets after you do your initial analysis might be really powerful and provide a unique insight into mechanisms of exhaustion and activation.
Yeah, that's definitely something I'm gonna have to think about. And just to tie, what you said back to Ben's point, where you said these are cells. I think you said these are cells that were not originally causing any pathology until this change was made. Which that kind of reinforces this this evolutionary perspective that this signaling that we're interrupting with, this signal that we're interfering with, it's not there for no reason. There was a purpose to it, and by interfering with it, we need to be cognizant of kind of the risks and knock-on effects that can be involved with that. So that's yeah, that's interesting. I'm gonna have to think some more about that.
Good. Okay. Well, it sounds like there's actually more interesting discussion, and so maybe we'll let Chris and his committee carry it on there. So thank you everyone for attending today. And as you saw from Chris's timeline, he'll be back at some point in the future to defend his dissertation. So see everyone later. Bye-bye.
Thank you.